Good morning. Good morning. Good morning. I hope that in your transit, if you've been outside already, if you're on campus, that you avoided the rain this morning. And if not, I hope you dry off quickly. Let me arrange these screens here. Keep letting people in. Come on in. Come on in. And thank you for finding very nearly all of those. You can finish this with the PDF later on. So pat yourself on the back. Well done. And let's get going. We've got an alliterative day today in 3700. Excuse me. And I hope no more of that. I will try and catch it and mute beforehand. We have an alliterative day. It's Monday. We're going to be talking about extending some of the life history theory and evolution of sex. So we get from the theoretical to some of the actual applied circumstance and rituals that we see with mating. And then we're going to be talking about molting. And molting ties together Monday and Wednesday, because we're going to be talking about respiration. And so the interface between the interior and the exterior of the animal is rather important. But before we begin, previously on Zoology 3700, we were talking about all of these things. We were talking about gonochoristic versus hermaphroditic, sexual and asexual pros and cons, bet hedging, competition, the Red Queen hypothesis, the role of parasites, that kind of thing. And then as kind of a, not a goodbye, because we're definitely never leaving this environment, but as a nod to kind of the first four lectures, looking at generalized life history strategies for particularly the marine and the aquatic environments, where we've been spending a lot of time in the first part of the course. Where are we going today? We're going to be talking about mating rituals and selective pressures behind that would perhaps initiate and maintain them. We'll talk about developmental modes in insects, because insects, the most diverse of the arthropod lineages, are going to be our in to thinking about mating. And so some of this is going to be picking up threads that we presented back in 2700 and extending those things. And why are we doing this? We're doing this because one of the outcomes that I would hope from today's material is that we and you can appraise and synthesize and interpret the diversity. Like, if we're going to talk about a diversity of mating systems, you have to think of, like, it's okay as a gateway drug to think about bowerbirds, to think about birds, and to think about visual things. But if you really are interested in sexual selection, oh my goodness, you need to be working with the invertebrates, because that's where the diversity is, and that's where the diversity of strategies are. And so then switching gears from mating to molting, we're going to talk about being able to synthesize and interpret examples and appraise novelty after we've spent our time together thinking about developmental modes used by insects, the holometabolous, ametabolous, hemimetabolous things, and then actually into some of the hormonal pressures and hormonal pathways that are happening for an insect during that period, and then some consequences of that. All right, let us begin. So these are some of in the vast diversity, and so granted, and I know I often start or include this near the start, but this could be a class. Yeah, insect invertebrate mating rituals, that could be a class. You could do, there certainly are classes and courses that you can take at higher level in behavior, and if they're going to interpret animal behavior properly, they ought to be just replete with examples about invertebrates. And so today we're going to talk about these 5 or 6 leading kind of exemplar candidates that you can either return with or enjoy being introduced to, or brush up on so that you can impress others in the soggy wet bus ride home later today. Let's talk about serenades. And when we're talking about serenades, we're talking about an auditory call. And one of the classic auditory calls that's
Give a little whistle.
one of the classic auditory signals that's going right now, and has been from the kind of the later, if you're from this part of the world, from the later part of the summer on until heavy frosts, are crickets. And there's all sorts of taxa, and we're only going to dive into one of these examples here. But the sounds generally produced by males are to attract females at a distance, and they're not nearly alone amongst the animal kingdom for doing that. But they also share then these so the pros that at a distance your sexual signal is traveling potentially further than a visually displayed one would be potentially. The con for serenading would be to that male that there would be differential sex-based selective pressures because of predators. And so if predators can track that sound, not only can the female return to your calling location to engage in a mating ritual with you, but predators can. And here's an example. So this is a Hawaiian cricket where the male on the left-hand side, number A, number A, right, letter A. This cricket is singing. It's making a song, and the song is designed to attract females to it, but it is also attracting a parasitoid fly that was introduced to the Hawaiian archipelago in the middle part of the 20th century. And the parasitoid kind of flies up that sound signal, that sonic signature, recognize it, flies towards it, and then basically once it's close, so it's flying up the signature, and then it diversifies its search pattern and begins circling down in a spiral pattern. Then it lands and it walks across and it lays its eggs. This is a tachinid fly, so it's laying its eggs on the body. And so these are not parasitoids as you may have been introduced to, like the hymenopteran parasitoids, which have a piercing ovipositor. Tachinids are especially kind of interesting parasitoids because they lay their eggs on food items or on the exterior of the body. But of course, with a parasitoid life strategy, this is the predator that is eating one prey item in its life. And so all of the eggs from this circling in fly here at sea are going to be eating that male. The male might reproduce before then, but it is going to be consumed slowly from the inside by the parasitoids that are developing on its surface or interior to its body. The interior can happen once the eggs hatch. Some larvae can embed themselves on the inside. And if parasitoid life history strategies interest you, you're not alone. And if you're watching any of the Alien series through your life, but also the Alien Earth series, those xenomorphs, there's just so much parasitoid biology that is replete within that series. So serenading and its consequences. So the pros being at a distance attraction and the advertisement of your sexual signal. And now we've got some visual ones. And a classic one, of course, I don't think that's playing for me, so it's probably, there we go, is the peacock spider. All sorts of visual signals. We talked in 2700 last year about visual signals in the waggle dance that honeybees use to orient their siblings to food sources. And we talked a little bit about some waving strategies that scorpions do. Peacock spiders are just one of the most incredible visual signalers, and there's some YouTube links there that you can scan and visit later on, because there's a particularly I enjoy a German researcher who has set most of these to music, in particular the YMCA songs. So you can go and, instead of listening to me, you can listen to the Village People as you watch these signals. So these are tiny little spiders in Australia, and the male conducts these basically, hey, look at me dances, while also, and what's not totally apparent in the video here, is there's the arm waving, there's the peacocking with the colorful display, and there's also a dance that's going on. So there's tiny vibrations that are being set up. And this is kind of an amazing set of a routine, a repertoire, a quiver of male strategies here designed to attract and entice the female. So there's the arm waving, there's the degree of colors that are happening on the back, on the cephalothorax of the on the thorax rather, not the cephalothorax, of the abdomen of the spider. And there's also the dancing and the drumming that's going on. And that dancing and drumming and the visual display and the proportion and the banding of black and white on the arms, all of that varies because peacock, or the peacock spider, as perhaps you know, but perhaps you don't, there are hundreds of peacock spiders that do species-specific dances and have species-specific displays across them. And so the vibrations are being set up concurrently with the visual displays. And one you might have, if you listened to CBC Radio and the amazing program As It Happens last week, you might have bumped into a PhD candidate from the Sanger Institute in the UK who was talking to the hosts of As It Happens about the some research that he's been doing as part of the team that he's a part of that's been looking at genome size and peacock spiders. And they're in the middle of this, but they kind of announce preliminary results that, so watch this space. You can read the interview and kind of listen to the show there. But their hypothesis is that peacock spiders, in addition to all of this display, have enormous amounts of what we used to call junk DNA, or DNA, so the majority of our genome that does a thing we might have presumed, but we just don't know what it is. Peacock spiders have an enormous amount of DNA. I'm not sure where they fit entirely in the continuum of from the most to the least, but they have many times more genomic DNA than we do. And these researchers think that this unraveling the secrets in this unknown DNA might allow the spiders to adapt rapidly and then diversify rapidly to new environments and be associated with the high number of species that are showing all of these incredible singing and dancing strategies. Now we're researchers, completely a separate branch from the singing and dancing researchers, from less of the theater kids to more of the engineers. The engineers, and of course you can be both, but the engineers in this case are looking at, for a biomimetic perspective, they're looking at the particular scales and patterns that are on the peacock spider. And you might think, looking at that display, which part of that scaly body would you look at? Look at the colorful parts, right? Well, these people were actually looking at the black regions along the legs, because they noticed that these black regions are produced. This is a percent reflectance here. This is for one of the species, the speciosus, and looking at the spectral range of the reds and the blues, and this is incredibly low. So they're reflecting. Remember we talked about strategies in the plankton about adopting and opening your chromatophores and kind of hiding from blue light. So these super black regions of the peacock spider, they're not about hiding. In fact, they're about accentuating the other colors. So the black and white patterns that are then called into tight focus when those black and white patterns wave across the colorful body, they make the bright colors that are already bright appear especially bright and luminous. And biomimetic engineers are interested in, so here's a look at, from a light microscope on the left-hand side, to various scales down to, there's my pun, that's my first pun of the morning, scales. So those are the scales on the back of the chitinous scales of the peacock spider, and various scales, fields of view from a scanning electron microscopic view of the scales. And biomimetic scholars are interested in this because it's a low energy way of potentially attracting attention to road signs or to advertisements, but ways that we would pump energy into to increase the luminosity that would then, if we're going to increase the number of lumens, we're increasing the amount of power that it takes to run that sign. And perhaps there's a way of that the peacock spider can teach us of just providing some contrast with these super black. If we can make these super black, if we can replicate these super black regions, we might get some colors that pop and use that. So serenades and dancing and gifts. Gifts are a natural thing if we're thinking about mating rituals, and given often male to female, but it's a nutritional gift in this case of one partner to the other to entice them to mate. And you know tons of different examples. Mormon crickets are a particularly famous one. Dance or robber flies, these flies which are still out and about. And so you might have seen some of these moving slowly in the morning this weekend because they're still out about. And we talked about these in 2700. These are the flies that have not a very large brain, but if you can zoom in here and take a look at the ratio of the size of the antenna to the size of the ommatidia, to the size of the eye, the compound eye. These are super hyper visual predators. And they're not about smelling and tasting their way around, at least their prey world. What they're about is seeing things and tracking them down. And we talked about it in 2700, and we made the comparison between this taxon of fly and border collies, or your dog. If you're throwing out a frisbee and the dog is starting from over here, and it is computing intersecting vectors to be, so it's not running at where the frisbee was, it's running where it's going to be, and it's adjusting its speed and its jump to catch that frisbee before it lands. And your dog's got a big, complicated, beautiful brain, and it's full of love for you, more so than many other people in the world. And these flies might not have as much love for you, and they have a much smaller brain, and they're doing the same calculations. So and that results in them being able to predate other flies with large compound eyes that are moving around, but they are doing it then to present as gifts. And here's a video from YouTube of a male fly that has caught and presented another fly as gift to a female, and that then has been allowed to reproduce. And you can see the eye sizes of the different and the ratios between male and female of these robber flies is more or less the same. I'll have another nuptial gift example in a second. But I want to talk about intraspecific chemical signals like pheromones. Then this is often apologies. I said I would try and catch that, and I will. So we've emphasized some of what the males are doing to attract. So here, these are pheromones that the female is. The female's choice is really, really important. So we have this scenario where all sorts of animals, maybe all, are using, on some level, using chemical signals to speak to each other. Okay, moths, ants, wasps, stink bugs, so hemipterans, we've got hemipterans, lepidopterans, hymenopterans, etc., meaning many, many, many more. So we have a moth flying out and about that runs into a plume of pheromones that it intersects. So it's doing a casting flight or a ranging flight that it then flies into and begins to turn back and forth. And this is somewhat analogous to that unintended auditory recipient, the parasitoid fly, flying up the sonic sound. But here's it's flying up. This is an intended recipient, the same species, flying up this pheromone trail until it casts about to find the most dense concentration of these signals. So the female, the positioning of, and this is an example of here, of a species, the males here and the females of Alicia lineata, which is a moth in Costa Rica. It lives in dry forest that looks an awful lot like this. And the positioning that the female assumes before she starts releasing this pheromone is really important. She positions herself moderately high in the undergrowth and then begins to pheromonally call males 3 or 4 hours before dawn. And she does that nearly right away after she's emerged. So this is she's emerged from a cocoon in her own life cycle. So I suppose we're linking the molting and the mating here. So she has molted, she has emerged out of her pupa, she's transformed, and then she begins that 3 to 4 hours of calling. And she calls from that point until sunrise, and males will then fly and do that ranging and casting to swim upstream in that pheromone trail. And she basically if a male finds and she accepts the male, she stops releasing the pheromone. And if not, she continues to call. Now, another strategy or another ritual involved in invertebrate mating is mate guarding, which we see in a lot of taxa, from soapberry bugs, which you can see outside around here, stone crabs, and cuttlefish. And this is the kind of behavior where males guard the female to minimize or prevent her from mating with other males after she has accepted him. So this can vary from short term to long term. I just said it can be post, but it can occur pre. There can be a male that anticipates being successful. So here we go. So there's our two cuttlefish here. We've got a female on the right and a male on the left, and they begin to mate. And when they're done, the male follows around the female until another male arrives. The first male then pushes its arms and makes lots of aggressive chromatophore, so lots of flashing and ink to dissuade that arriving male. In this case, it wasn't successful. The second male was successful. And that then links into elements of choice in terms of which that there can be across all of these species in terms of which sperm are accepted. There can be apparent copulation but not reproduction, and this is part of why taking a full course on animal behavior that is replete with invertebrate examples will be very, very interesting. Caramel appears, our 20-year-old Caramel appears and reminds me to take a look down at the chat, but I don't see anything there yet. So we'll keep going. And we'll talk about another kind of a mating ritual that we definitely did talk about in 2700. And this is the hypodermic insemination that we see within some of the hermaphroditic flatworms, marine flatworms. So these are animals that possess both male and female genitalia, and they tend to fight. So the penis is one that can pierce, there it is there, the body of the other in this dance. And from an evolution, what evolutionary theory or life history strategy has taught us about these dances in the past is that in as much as these flatworms want anything, the race here is to impregnate the other first, because the one who pierces with its hypodermic insemination, the other one then begins to produce eggs, and that takes more resources for them and prevents them then from dancing in this competition again with other members. There can be reciprocal insemination, so they both end up having to having to generate eggs. Now, we talked about nuptial gifts earlier with the flies. I've got another example here with spiders that you might have bumped into in the past, and these are, so in the same way that the robber fly will catch flies on wing, that of course spiders, what do we know about spiders? I know, well, you could sing songs about them, about people that catch them and they swallowed a fly, so they swallow a spider that was wriggling in. Let me know in the chat if you're still there. So I think you saw that warning that popped up on my screen saying the disk is full, but I just want to make sure you can still hear me and it's not so full that this transmission has ended. Can you hear me? Hooray, thank you. All right, so the male, the spider, catches flies, binds them, and presents them to females to initiate copulation. And if rejected, he might rewrap and present the gift again. He'll re-gift the fly that he's given. And the gift that is given, one of the things that you might have bumped into with spider reproduction, thank you all, is that females often end spider reproduction by eating the male. And so the hypothesis, the mechanism here that is presumed is that by having a male spider that is giving you a gift, it makes it much more likely that the male spider will live and survive. Some spiders, like this one, the Mira, the Surina Mira, it goes a little bit farther and it presents the gift. Once, if the female starts to eat the gift, it then wraps the legs of the female up as she's eating, so that it can copulate with even lower chances of being consumed by the female. And again, a touch back to 2700, where we talked about the hermaphroditic snails that have a dart sac and a love dart. And again, so we're looking at different species of snail and different morphologies of these love darts, where there's internal fertilization and a dart sac, and the courtship can last many, many hours, but this calcareous stylet delivers a spermatophore that is received in a particular region of the body called the diverticulum it's kind of a small canal, if you remember, and that contractions of that canal can then increase the concentration of the sperm and then close it and prevent further mating. And if you don't remember it from 2700, you probably remember it from Z. Frank. And so let's have a look at their presentation.
When they mate, land snails circle around one another. Think full-body French kiss, but with more mucus. As they do this, they're looking for opportunities to impale each other with their love darts. I wish this was a metaphor, but it's not. They actually have love darts. Like actual darts. It's crazy. Land snails have a totally different level of kinky. These calcified darts, which look totally badass, are stored in the snail's dart sac, located right below the penis. When the darts impale the mate, they inhibit spermicidal secretions, which makes the next part more effective. After a spirited darting, the snails insert their penises into each other's vaginas, and then they just hang out.
There's a little collembola crawling across that canal there. So, yeah, tip of the cap to Z. Frank, always.
When they mate.
Okay. All right, there's my reminder from Caramel to check in, and thank you for confirming. Yes. And this is our transition. So these are the mating rituals, and for the next kind of 20 minutes, we're going to talk about some specifics as a reminder about different developmental strategies in arthropods in particular, but when we're talking about molting, of course, we can include the aquatic lineages and crustaceans. So within insects, we can have we see three different kinds of development across the insect world. We see ametabolous development, where you get slightly bigger from an egg once you transform into a small thing, that small thing form. We see this in collembola, for example. So hexapods, not even insects, but hexapods, right? Then we have hemimetabolous development, where eggs to the first stages and these nymphal stages look slightly different and perhaps don't have wings, and then we have a final jump to wings. And then in the holometabolous lineages, like iconically the butterflies and moths, eggs to larva, numerous instars of larvae that get bigger, and then a refractory period or a pupa where a large transformation is going to occur from the crawling caterpillar and the herbivorous caterpillar to the adult. So there's the ametabolous again. So we got Thysanurus and collembola given as the example here, where there's no change in form, but an increase in size at each molt. And that's a critical part of the molt, right? It's not just thinking about the butterfly from a caterpillar. I'm going to be a beautiful butterfly. It's not just that big, incredible transformation at the end. It's also just about size, about allowing the organism to get bigger. Then we got hemimetabolous, where there's little change in form. You get bigger and bigger and bigger. And then the final molt, we see the development of these wing pads. And we see this in the odonates and the orthopterans and the hemipterans. And the hemipterans was the last example. That was kind of a soapberry bug, and this is a grasshopper. And then the holometabolous. So the larval instars, once you go from egg to larval instars, they're similar. But then in the final stage, there is a dramatic change through the pupal stage to give you a winged adult. And we see this in Lepidoptera iconically, but we also see it in beetles, we see it in flies, and we see it in Hymenoptera. And if we were to look at this, as we did in 2700, from a phylogenetic perspective, across the hexapods here. So we've got some crustacean outbranches here, but we've got hexapods across. And so for the entognaths, where the jaws are recessed, those were the Columbella and the proturans. These are the ones, these are the ametabolous lineages. And then we have a non-monophyletic group of the hemimetabolists. So we've got odonates and ephemeropterans. These are hemimetabolous things. So there's a change. They get bigger and bigger and bigger, and then there's some wings being added in at the end. But we also see it in some of the hemipterans here. And then we see the ultra successful lineages of the holometabolous lineages, the flies, the Lepidoptera, the beetles, and the Hymenoptera here that have instars that increase in size, and then a pupal stage where really you change your job. And so looking at that blue bullet at the bottom there, this we think has one of the reasons, the mechanism behind the success of this strategy is really about resource partitioning, that it avoids competition within lineages, within species, within families, because there are different and complementary roles in the life cycle. So adults and immature adults and the larval instars, the caterpillars, are eating and doing very different things. And in fact, for much of the diversity of life over the last 150 years, if you've been interested in these kinds of holometabolous lineages, there remains an enormous challenge taxonomically to resolve which larval stage, the instar, goes with which adult, the ones that can look dramatically different. And that is true for butterfly, for caterpillars and moths. It's true for ants, where we would often see, you might see not necessarily juvenile or pupal ants, but you would see workers and reproductives that would be flying around and not know which, or be able to resolve which is which. From a evolutionary perspective, we think that the ametabolism, that mode is the ancestral, and that these are both derived. The holometabolous lineages are monophyletic. The hemimetabolous ones is a description, and it's not monophyletic. And there's just a look through here. So we emphasize the butterflies and moths in the past, and the video kind of loops through an iconic example from our part of the world of the monarch. But then we've got the same kind of thing that's happening in tree stumps all over the dairy bush and the rest of the arboretum where you were last week, of these C-shaped beetle larvae that are going to get bigger and bigger and bigger and bigger until they're the size of your thumb, your little finger, and then they're going to pupate, and then they're going to emerge as an adult. So with the ametabolous lineages, they grow in size, they directly develop, and so the only real difference between the adults is the onset of sexual maturity and size. And those are the lineages, we've talked about the lineages we see it with. So we talk, one of the phrases that as an invertebrate biologist or an insect, an entomologist that you might run into are nymphs or naiads. These are just what the immature instars are called by history. And so nymphs would be terrestrial. So if we saw these hemipterans, these bugs around these, we would call nymphs or different instars. And if we saw it with aquatic bugs, we would call them naiads. And they resemble the adult in form and feeding. And if the early stages tend not to have wings. And then finally, with the holometabolists, the incredibly successful holometabolists, that we've got this in pupal stage that really changes everything. It changes the ecology and allows, and then indirectly through time, the evolution, because you can imagine that a caterpillar itself is not a great disperser and a connector of populations. But an adult in this case will. And this is kind of the opposite of what we would see in a marine system, like you're working with in your invertebrate example, where the adults are more sedentary, and it's the juvenile life history stages that we invoke dispersal for, to recolonize populations that have gone locally extinct, or to colonize new and emergent open areas. So the pupal stage, just as a final word about it, this is that break stage, this beautiful one here between immature and mature, where the larval structures are basically homogenized and broken down, and the adult structures are formed. And this it's controlled as so much as in our life is by hormones. It's a non-feeding, usually sessile stage that can last weeks or months or years. And that hormonal stage, I'll check in with the chat for a second, but it's that hormonal stage as it affects molting that I'd like to check in briefly with for the rest of today. And I don't see any new questions there. All right. So when we're talking about molting in insects or hexapods, we're talking about the cuticle or the integument, synonyms, right? So the cuticle is secreted by the epidermis, by this basement membrane, the outside cells here. And it's laid down in several different layers, but we can talk basically about this layer, exo, meso, and endo, as being the procuticle that's comprised mostly of chitin, and then the epicuticle, the outermost layer that is a waxy layer predominantly, and has a lot to do both with sexual signaling, but also with species-specific signals, and also with water retention. And so looking for beetles like this. This is an Aleocaran, or it's not an Aleocaran. This is a Nautilus beetle, a staphylinid, a rove beetle from Costa Rica. And that video is looping through a sifting tray of mine as I sit in the just beneath the cloud forest, so high elevation rainforest in Costa Rica. And what the camera's zooming in on is this figure here. This is an unnamed species of a nautilus. This is looking at it here. And what I wanted to do for a second was to show you it in person, to say, yeah, so that's about 1 millimeter long, that organism is. But you can also then, so that you can appreciate that things that look small and brown and invariant at our big clumsy stage look very, very different when you can look more closely at them. And those species-specific differences are often at a cuticular or an integument level. So this is scanning electron microscopic images that we've taken of rove beetles from those tropical forests in the facility in the science complex at Guelph. And so we're looking at, I think, two things. One of them is that if you haven't seen scanning SEM images of beetle chitin or insect chitin before, I want you to reflect on the differences between, these are all different species, but that how hairy they are, that beetles are or that insects are often very, very hairy. And those hairs can be very complex. They can, and we'll return to this because they can often be about receiving proprioceptors or receiving sound or vibration. And they can be densely or less densely arranged on the body. And that the scale, that the integument itself, even though it's often talked about as armor, when you look at it up close, you can see that even a shiny insect like a beetle has scale-like structure in its integument as well. And there's a zoom in at some of the hairs that have been broken off, but at the features on the surface of that beetle elytra. Now that surface can change radically. This is a different species where the surface is less scale-like and is more sculpted, almost looking like a golf ball. And then one in particular that I like to finish with because it always reminds me, I forget, the cape. The cape has a Star Wars Imperial kind of Darth Vader-y aspect to me. It evokes that kind of feeling in me. And what you're seeing here, these bristles, these are the wings that, of course, so rove beetles are the beetles with a very short elytra that do the kind of incredible origami, more so than any of the other beetles. Underneath the elytra, and the elytra in the beetles is that modified wing that becomes a cover and a hatch. And if you're Minecraft literate or grew up playing Minecraft, of course, you know that. So when we're talking about the integument and we're talking about growth and molting, we have to think about all of these different layers. And this is a quick refresher of some of the materials that you saw back in 2700. So we have the basement membrane and the epidermis that's secreting the procuticle, the exo, the calcified, or the uncalcified, but the chitinous, the tough cuticle, and then the epicuticle up at the top. And we've got hairs and setae with different glands that opening. And you saw examples of all of these features in those last images. And when we're thinking about molting, the diagram is going to switch to this one, to a cross section, to looking at these ecdysyl lines. So this, revisiting 2700, this is 3 different stages in growth. So A is most of life for this particular insect. Tissue growth is normal, and we have endo, exo, and epicuticle being secreted by this epidermis. But then we have ecdysis, ecdysis, and post-ecdysis. So this is when the epidermal cells are stimulated to start secreting molting fluid. And the molting fluid concentrates along these what look like little gray mountains, these ecdysyl lines. And then that's where the split happens. And the split happens to allow the now larger organism to emerge from its constrictive case, to inflate itself bigger, and to continue its life. And you've seen this all in your lives, but if you're lucky enough to bump into these cicada casts and cicada skins on trees around your yard or around your parks or around where you're living, you've seen that. Now, what's happening here, and what this is an early instar version of our oldest son, but he found this particular one in our yard. And as we took pictures of it kind of emerging and inflating its wings, so there's the uninflated wing. Now it's pumped up. It's about an hour or so later. But you can see these tubes heading out. This is a reminder that the trachea, the breathing apparatus that insects have, is also their exoskeleton, right? And so as they're molting, this period of respiration, or this normal respiration, is interrupted. So molting for many insects is not just dangerous because of you're softer and you're not mobile, but it's also because you can't breathe as well. So many insects, it's a real interruption. It's a necessary interruption to normal life history stages. So from an environmental or a physiological perspective, we have, and there's a lot, I'm going to walk you through all of these lines here, but we're basically tracking to get to the production of this steroid, the ecdysosome, that is going to stimulate molting, that molting fluid. We have an environmental stimulus, and this can be all sorts of things. It can be seasonal. It can be the size, the growth and tension on proprioceptors in the exoskeleton. But that is received by the brain. Whatever it is, that environmental stimulus is received by the brain. Then the corpus callosum produces PTTH, that then stimulates the prothoracic gland. And the prothoracic gland, farther down the length of the body, is what produces the ecdysone. And the ecdysone, which is spelled correctly here and not in the bullet, is what then travels through the body to the epidermis and stimulates the production of those, filling up those ecdysyl lines. And so as you look through the life cycle of a particular instar, so here we've got like a cockroach, say, and we've got instar 1, and this is time on the bottom, and then we've got juvenile hormone that is also needed with the ecdysone, but the level of ecdysone is shown in black because that's what I'd like us to focus on here. And to see, and the black line, so this is the ecdysone, and this is the point of molting. And what it's showing you is that the molt is always preceded by a surge of ecdysone. And so this is a hemimetabolous lineage. And so we've got kind of similar sized peaks, or this repetition until you're an adult, okay, until you got that final stage. Now, when you're a holometabolous insect, the peak size can change, and you can see that so you get different peaks with different instars. But then the big pulse that is needed in the pupal stage to transform the immature to the mature form. Now, so these are the signals within the body determined by different environmental stimulus. But what I would like you to think about for a second and throw out in the chat here is what effect would have of stimulating an early molt? What effect on the insect would there be if molting was stimulated early? So if some reason this peak occurred here, if we shifted the peaks earlier, what would happen? What might happen to the animals? Ah, great. I see some thoughts already. Let's pull these up. A smaller adult size, potentially, if they can successfully transform. What's more likely is death. Mutations, slower development. So it wouldn't actually be slower, it would accelerate the development, and it might result in the softer and less developed exoskeleton might be one of the consequences that leads to death, that the rest of the body is not ready for this developmental leap. And so strangeness, certainly, and I think some of your answers invoke that. Death, potentially. Now, in 2700, we talked about different insecticides that people have tried to develop that mimic ecdysone and stimulate molting in mosquito species that vector malaria to try and get them to molt early when they're not ready, that can then result in its death. And the example that I wanted to talk about here to diversify our thought of the ecdysone environment and how it can be manipulated by things in the environment is by predators. And so we talked about parasitoids here. This is a hymenopteran parasitoid working with a fly. Now, parasitoids being parasitized is stressful, and it can result in nutritional deficiencies because everything you're eating is being actually eaten and consumed by the developing larva inside you. So we've got a normal spider making a normal web, but then we have a wasp, a parasitoid wasp, attacking a spider that is parasitized. The larvae begin growing inside of it. And then what researchers have noticed is that parasitized spiders begin to build modified webs, strange webs, until they're killed by the larva that pupates in that web. And so what they're thinking is that the fluctuations in the steroid, because ecdysone has a strong association with stress. And so fluctuations in stress, remember the environmental stimulus, that's one of them that can prompt boosts in ecdysone that can then result in pre-molt behaviors or weirdness. And you talked about soft or less developed things. That's essentially what's happening here with this spider. So we have a pathway that researchers are thinking that parasites infect hosts. And then we move along here on the right-hand side to see the production of different kind of stresses like low nutrients, an immune response, adaptations or effect on the central nervous system of the host that then boosts the production of ecdysone that then results in pre-molt behavior. So in the case that we talked about from the humans using this strategy, the death pre-emergence of the mosquito, and here it's just pre-molt behaviors that are out of time but can benefit the developing hymenoptera, the developing parasitoid larva to the cost of the spider. All right. Now, so that's our alliteration period. We've got mating rituals and developmental modes and metamorphosis and molting. Where are we going to go on Wednesday? We're going to keep talking about the cuticle and integument. We're going to talk about gas exchange and circulation and what you to be working on. Well, first of all, a reminder that this is your off week in the lab. So there's you can think about your class experiment, and there's some details that Shayla has posted online. But you can also then don't forget about your invertebrate R assignment that's due on Friday, October 3rd. And if you haven't already, it's a great chance for me to remind you to make some study groups for your case studies. And that's about it. Be good to yourself, be good to each other. I'm going to stop recording here in a second. And I'm going to hang out for some questions. So